All right, welcome everyone. Thanks for coming to the last IDFE seminar of the year. Juan will do the introductions in a minute, but I just want to thank everybody for being faithful attenders, or not faithful attenders in some cases, but for attending. And particularly to thank Juan Gago, Julia Dykman, who's over there, and Salome Kuchukidze for organizing this series. It's a lot of work. It's fun work, but it is a lot of work, and they have done a great job. Without further ado,
Hi everybody, we're coming today Erin Mordecai. She's an associate professor in the biology department at Stanford. Her research focuses on ecology of infectious diseases. She's interested in how climate, species interactions, and global change drive infectious disease dynamics in humans and natural ecosystems. Erin finished her PhD in 2012 at the University of California, Santa Barbara. She then completed a 2-year NSF postdoctoral at the University of North Carolina, Chapel Hill, and North Carolina State University, and she has been at Stanford since January 2015. Thank you for being here today.
Thank you.
Okay, how's that volume? Can you hear? All right, excellent. Thank you so much, Juan. Thank you, Mark. Thank you for all the students and postdocs who are on the organizing committee. It's really a special honor to be invited by students to come. Are we okay on sound? Okay. So I'm excited to share some research with you today that my lab has been doing recently, thinking about climate change impacts on infectious diseases and going beyond kind of looking at average warming. Before I get started, I want to take a moment to thank the many collaborators and funding sources that have made this work possible, including NSF, NIH, several of the Stanford Institutes and Centers, and some people at the Peruvian CDC, Stanford, Berkeley, and some other collaborators who contributed to the work that I'll present today. So as an ecologist, I'm really interested in understanding the way the world works, how species are interacting with each other, how those species interactions drive the dynamics and evolutionary trajectories of species, how those species interactions lead to emergent properties like competition and coexistence of diverse communities, as well as processes like the spread of infectious diseases and predator-prey interactions. And I'm also interested in how these relationships are changing over time. And these are tricky things to study because they're part of complex ecosystems. They're really interdependent, really tangled, really complex, really nonlinear. And of course, humans are arguably, committing one of the largest and most poorly controlled experiments of all time on ecosystems. We are transforming the planet in many different dimensions. Humans have cleared over half of the world's tropical forests. We've blocked over 60% of the world's free-flowing rivers. We've converted most of the world's arable land to agriculture already. We've contributed to the 6th mass extinction, and we've warmed the planet already by over 1 degree Celsius and counting. So humans are contributing to these multivariate ecological and environmental changes, and I think these aspects of anthropogenic change are the defining societal and scientific challenge of our time. There's many different lenses and many different dimensions to this anthropogenic change, but the one I'm most interested in is how global change is affecting infectious diseases. This is fundamentally an ecological and an evolutionary question because it involves interactions between species and evolutionary processes that go on within those species. And because of that, it's a challenging question to address. How do we really understand how global change is affecting infectious diseases when we know that any given disease is likely to be embedded in a complex ecosystem where there's multiple hosts, potentially multiple vectors, there's interactions between those hosts and vectors, there's multiple parasites that might be interacting at the individual host level or at the population level, there's multiple environmental drivers that are affecting both the parasites and the hosts and their vectors. And so all of this leads to lots of complexity. There's lots of different interactions here in this diagram. And there's lots of opportunities for nonlinearity, where a simple incremental environmental change doesn't necessarily result in a simple incremental response in the infectious disease system. So in the context of these complex systems, how do we study infectious disease dynamics as they're responding to global change? A lot of the approaches that we've adopted in my work involve using data-driven modeling, a lot of times mathematical modeling, sometimes statistical modeling, but the goal of this modeling is to allow us to integrate lots of different types of data that allow us to link processes across scales, from the molecular and organismal all the way up to the population and community and even global scales. So today I'm going to focus on our work on vector-borne diseases. These are pathogens transmitted by biting arthropods like mosquitoes or ticks or flies or fleas. These are important from a public health standpoint because they include diseases like malaria, dengue, yellow fever, West Nile, Lyme disease, lots of the very high burden infectious diseases out there. They're also really interesting from an ecological and evolutionary standpoint because they require these vectors, which in turn links these diseases back to the ecology of the environment around us. Today's talk is going to focus on the impacts of climate change on vector-borne disease transmission. And I'm going to start by giving some brief background on some older research that we did, 10 years ago or so at this point, that kind of motivates the work that we're doing now. And from that research, we'll identify 3 key gaps that I want to address today. So starting with some background, we can take a trait-based approach to studying the transmission of vector-borne diseases. Thinking about a mosquito, we can think about its life cycle. So how might a mosquito's life cycle and transmission cycle be affected by climate change? Well, mosquitoes lay their eggs on the side of a body of water. They hatch and go through 4 larval developmental stages. They pupate and they emerge as adults. So survival, fecundity, and development rate are important for the mosquito life cycle. Of course, here's the part they're famous for. The female mosquitoes take bites of blood meals from vertebrate hosts, and they use those blood meals to produce eggs. So that biting rate is also responding to the environment. And then the parasite, for its part, has hijacked this mosquito life cycle so that when that blood meal that the mosquito feeds on contains parasites, instead of just getting digested and excreted out of the body, it instead is able to break through the midgut barrier, disseminate throughout the body, and bind to the salivary glands. And that's when the mosquito becomes infectious to the next vertebrate that it bites. And so this process of extrinsic incubation and vector competence is also dependent on the environment. So if we look at how temperature might affect the transmission cycle, we can imagine that temperature, in particular, would affect every aspect of the transmission cycle because the mosquito is a small-bodied ectotherm. So its body temperature is always matching the temperature around it, and its biological processes are all acutely sensitive to temperature. So thinking in this kind of trait-based way, we can then say, well, how would temperature affect transmission of a mosquito-borne disease? We can piece together all those different impacts across the life cycle. And that's what we've done in this previous research. So we can go into the literature, and we can look at studies where entomologists have measured different mosquito and parasite traits that contribute to transmission across experimental temperature ranges. So that's represented by these circles here. We can find those experimental data and then fit continuous thermal performance curves for each of these traits of mosquitoes and parasites that contribute to transmission. So then we get direct empirical relationships between temperature and mosquito and parasite traits. But then we want a way to synthesize that and really bring it together to understand how the entire process of transmission is responding to temperature. And for that, we can use a model of R0 versus temperature. So R0 we can write down as an expression that's nonlinear, but it's a fairly simple function of these traits of mosquitoes and parasites that are themselves nonlinear functions of temperature. So this gives us a way to sort of have a static snapshot. We're not talking about the absolute magnitude of R0 here. We're talking about where are the absolute limits where R0 must go to 0, because one or more of these traits is 0, and what is the thermal optimum for R0. And we can calculate this relationship as a function of these empirically measured vector and parasite traits. We can then validate this lab-based theoretical model of R0 versus temperature against field-based data on transmission or incidence of infectious diseases versus temperature. And then with that validated model, we can use it to project current and future transmission risk as a function of temperature. So we applied this research method across a variety of different important human mosquito-borne diseases. It was published over a series of papers, but those were summarized and synthesized in this one Ecology Letters paper, including malaria, dengue, chikungunya, Zika, West Nile, Rift Valley fever, and many other mosquito-borne viruses or pathogens. So overall, what we find in this body of research is that we see nonlinear relationships between temperature and each of the mosquito and parasite traits. So here's an example for the larval development rate for Culex quinquefasciatus. You can see that it has a nonlinear relationship with temperature, which has an intermediate thermal optimum around 30 degrees Celsius and declines to 0 at both warm and cool temperatures. we see this type of relationship consistently across all the different vector and parasite traits that we looked at, across all these different major vector species, including the vectors of dengue and yellow fever, the vectors of West Nile, and the vectors of malaria. So when we put together our temperature-dependent R0 models for each of these systems, we see this really consistent hump-shaped relationship with temperature, where transmission declines to 0, so there's 0 potential for transmission at some critical thermal minimum for each of these. It then increases up to an optimal temperature and then declines up to a critical thermal maximum above that optimum. Question?
Sorry to interrupt you.
Sure.
Given the fluctuations of temperature over
Yeah.
the day, is this given a mean temperature? Does it vary depending on
Yeah, good question.
how much temperature is
Temperature fluctuations can have a big effect, particularly when you're looking at these like marginal temperatures. You can do rate summation approaches where you basically take each of these constant thermal performance curves. So all the thermal performance curves are derived from constant lab experiment temperatures, and you can integrate over these to predict what would be the performance under variable temperatures. And I think there's still some research to do to really validate that that integration works. But as I'll show you in some future work, even with the simplicity of using constant temperatures here, we managed to capture some pretty big field patterns of transmission, even though temperature variation is likely to have nonlinear effects. But that's a good question. So what we see here is that across these different mosquitoes and pathogens, they all have a hump-shaped relationship with temperature, but their thermal optima and thermal limits are different. So if we zero in on some of the most important pathogens, we have dengue and Zika with the warmest thermal optimum around 29 degrees Celsius. We have malaria peaking at 25 degrees Celsius. We have West Nile and Triple E peaking at lower temperatures like 23 or 24 degrees Celsius. So just based on the temperature dimension of climate change, what does this imply for how climate change might affect these diseases? It implies that if you're starting out at a pretty cool temperature, any warming is likely to increase all of these pathogens to varying degrees. If you're starting out at a fairly warm temperature of 29 degrees and above, any further warming is only going to decrease the potential for transmission. And then there's an interesting range of temperatures here in the middle, between about 24 and 29 degrees Celsius, where the same change in temperature might have directionally different effects on different pathogens, such as malaria going up and over its thermal optimum, dengue rapidly increasing, West Nile rapidly decreasing in this same temperature range. So these are kind of the foundational studies that motivated the work that we're now doing going forward. What we found here is that the trait responses to temperature are nonlinear. The thermal optima and limits vary across different taxa as well as across different traits. And that because of this, climate change is predicted to drive shifts in suitability rather than widespread increases across all pathogens, because we may see some pathogens actually exceeding their thermal optima as the climate is warming. But it's also important to note that all of this is mediated by human activities. So temperature, of course, is not the only driver of these vector-borne diseases. They're very complex systems. So the next question that this brought to mind for us is, do these trait-based approaches really capture the full complexity of vector-borne disease systems? And specifically, how do we predict the quantitative impacts of warming on transmission in real-world complex systems? So not just relative R0, but how many cases do we get? How many additional cases do we get of a disease per degree of climate warming? Second, how does extreme weather and weather variation more generally affect transmission? And third, how will mosquitoes adapt to climate warming? So there are other gaps, but these are the 3 gaps that we chose to focus on first for this work. So first off, trying to quantitatively predict the impacts of warming on human disease incidence. For this, we can sort of use our trait-based approach as a hypothesis generation. So we have this expectation of how temperature should be affecting each of these diseases. But we can then leverage some econometric causal inference tools that are developed to do causal inference on observational data sets, particularly when you have a very strong hypothesis, mechanistic hypothesis about how the system is working. And the way these approaches work is they leverage quasi-random variation in a system in order to quantify the impacts of environmental change. So I'll walk you through what that means. Here we're asking, how does temperature quantitatively affect incidence using a causal approach from econometrics? We're going to ask this question for dengue, which is transmitted primarily by Aedes aegypti, the yellow fever mosquito, with a secondary vector, Aedes albopictus. You can see here, these are the R0 thermal performance curves that I just showed you for these 2 species, where Aedes aegypti has the warmer thermal optimum. Dengue has a really broad distribution throughout the tropics and subtropics. It's a febrile illness. I'm sure you've all heard of it probably. And this is kind of an estimate of its force of infection across its range. And dengue burden has been really rapidly growing. Since the 1970s, when it was sort of at its low point, it's really reinvaded a lot of the Americas and Southeast Asia. And we saw our biggest dengue years on record in 2023, followed by a doubling in 2024. So this is a really rapidly growing problem throughout the world, and particularly the Americas. So how does temperature affect dengue incidence? And then from that, how have historical climate change and future climate change impacted dengue burden? And this is work done by 3 members of my lab, Marissa Childs, who actually went on to do a postdoc here. Some of you might have met her. Mallory Harris, who is now a postdoc in Joshua Weitz's group at University of Maryland, and Kelsey Lieberger, who's now a professor at Arizona State. So what we're doing in this work is we're leveraging natural variation in temperature as a kind of quasi-experiment. So this is the way our approach works. We use panel data, where you have observations of dengue incidence over many different spatial units over many different time periods. And we're comparing each of these units to itself over time while controlling for various sources of confounding variation. So observable variables like precipitation or population density, as well as unobserved and unobservable variation. And that's where comparing units to themselves over time is helping us control for some of that kind of unobserved variation that might drive dengue variation. And importantly, we're testing a specific hypothesized causal effect. So this is not like a lot of models in biology where you're saying, what explains the most variation in dengue? Here you're saying, what is the effect of temperature on dengue incidence while controlling for as much other confounding variation as I possibly can? So the way this approach works is we have dengue incidence as a polynomial function of temperature because we expect it to be nonlinear, so we want to allow some flexibility in that shape. We assembled a large global data set of dengue incidence from 21 countries, over 6,000 spatial units within those countries, averaging 11 years, but some of these time series go back as far as 30 years. We matched each of those dengue observations with temperature from ERA5 data from the same month. We also matched these with precipitation. And then we include what are called fixed effects in econometrics. This is the part where you're comparing each unit to itself over time. So you have a fixed effect for administrative unit. So, a given place might just have higher dengue on average. That variation is subtracted away. We have a country year fixed effect. So a given year might have been particularly bad for dengue in Brazil, for example. That variation is subtracted away. And then we have country month effects. So these are kind of controlling for the shared seasonality within a country. So now we're looking at these kind of tiny deviations from these averages and saying, do those years that had slightly higher than usual dengue incidence also have slightly higher temperature? That's kind of the variation we're leveraging here to estimate this temperature-dengue relationship of interest. And then we're fitting a population-weighted Poisson regression. So the units that have higher population sizes are contributing more to our estimate. This is what the data set looks like. So we were able to get data at the sub-country level from most of the countries in Latin America and the Caribbean, as well as parts of Southeast Asia. You can see some important data gaps here, which I'll get back to. But what we're showing in this two-dimensional color plot are kind of high temperature in red colors, high dengue in blue colors, and high temperature and dengue in the purple colors. So you can see there's a lot of variation in this data set. And then this is just showing some of the time series aggregated at the country scale, just so you can see how far back some of these go, as well as how variable the dengue dynamics are in different places. So this is kind of the raw material for the model. This is what we estimated for the temperature versus dengue incidence relationship. So here's temperature on the x-axis, relative log dengue on the y-axis in incidence. Each of these vertical bars is the country-level mean temperature, just kind of showing you where the different countries fall on this temperature range, with Bolivia and Mexico having some of the cooler temperatures, Brazil, Indonesia, and then Cambodia has one of the warmest temperatures in the data set. So this is our statistically estimated relationship between temperature and incidence from human case data. We can now go back and compare this with our predicted R0 versus temperature relationship. And we can make a couple of observations. First of all, they're both nonlinear. They both kind of have this hump-shaped, concave down shape. Second, we predicted from the R0 model that dengue transmission would peak between 26 and 29 degrees Celsius, depending on which vector was transmitting it. And we can see in this inferred statistical relationship that the peak is at 27.8 degrees Celsius, so pretty close to what we predicted mechanistically. We can also see, if we look at this empirical relationship, that the fastest rate of change of dengue with respect to temperature is occurring around 15 to 20 degrees Celsius or so at this accelerating part of the curve. And that maps onto the portion of the R0 curve that's also accelerating most rapidly. So together, this is kind of giving us support for the idea that these mechanistic trait-based models can really capture real variation that's going on in the field, despite some of the simplicities of this type of model. So now we can take this model and use it to project both forward and backward in time under different counterfactual scenarios of climate. So first we can look backward, and we can say if humans hadn't emitted the amount of carbon we have into the atmosphere, what would the temperature have been? And what would dengue have been given that alternative temperature scenario? And when we do that, we find that warming has already dramatically expanded the burden of dengue. Specifically, we attribute that anthropogenic warming is responsible for 19% of the dengue burden on average between 1995 and 2014, so not even going all the way up to today, within these countries that were part of the study region. That's the average, is 19%. But if you look at this map, you can see quite a bit of variation in how much of the dengue burden is attributable to climate warming that's already happened. And in particular in Latin America, in the high elevation parts of Mexico, Colombia, Peru, you see much larger shares of burden that are attributable. That makes sense because these are the places that are starting at cooler temperatures, as well as southern Brazil here. So now we can do the same thing but looking forward in time using simulations of future climate models. And we find that future warming is expected to also dramatically expand the burden of dengue within these endemic countries. So we project that warming is going to increase dengue burden by 61% on average across these study regions, with many regions facing more than double dengue burden because of climate warming by mid-century, so by 2050. So in particular, if you look at these circles, these are the high population centers within the data set, and you can see that especially in Latin America, a lot of those occur in some of the cooler parts, the higher elevation parts, where warming is expected to more than double dengue incidence by mid-century. So if we calculate just as a very simple metric the number of people already living in those places, we predict that 257 million people are already living in places that are expected to more than double in dengue incidence by mid-century. So this is some of the first climate attribution for infectious disease work that's been done, and it's showing really dramatic, large impacts of climate change that have already happened on dengue, and even larger ones that are projected to happen in the future. But we wanted to also compare different scenarios. So this is the SSP 3-7.0 scenario, which is kind of the business as usual scenario. If we compare that to the sort of better case, best case scenario of if we're able to really drastically reduce carbon emissions now, we also see that mitigating carbon emissions will have a major benefit in terms of reducing future dengue cases. So here we're comparing those two scenarios. We can think of these as like the worst case versus best case scenario. And across, our overall estimate is that it's about 18% better under the lower emission scenario compared to the upper emission scenario across the whole data set. But you can see that those benefits are much larger in some of the cooler countries. So here are the countries ordered by their temperature. So you can see in places like Bolivia, Peru, and Mexico, those benefits are very large of carbon mitigation. The benefits get smaller as we get into the warmer countries where there's less warming needed to get you into the optimal space for dengue. There's also quite a bit of variation around these estimates because there's within-country variation in temperature, as well as uncertainty in these future climate scenarios. I just wanted to highlight also that this approach, we view it as actually pretty conservative. For one thing, it doesn't incorporate future range expansions of dengue into places where it's not already endemic or hasn't been for a while. It also doesn't incorporate any country in Africa or India because we just didn't have data available that was usable for this kind of experiment. It also doesn't incorporate future uncertainties, which are just difficult to account for. Things like changes in control measures, future vaccines, potentially vector species expansions, and so on. So this is sort of our best estimate given the current status quo. So we find in this project that causal inference validates our mechanistic model predictions for how temperature is affecting dengue transmission. We find that warming is dramatically increasing dengue incidence, particularly in the cooler regions that are between about 20 and 25 degrees Celsius, where incremental warming is really moving them steeply up that thermal response curve. We find that historical climate change that's already happened has already dramatically increased dengue incidence. Future climate change is likely to make it even worse, particularly in some of those places that are already facing climate-driven increases in dengue. And climate mitigation is critical. So it's, at this point, not possible to prevent future temperature-driven expansions, at least based on climate mitigation. But by reducing carbon emissions, we can have substantial benefits in terms of averting future climate-driven dengue cases. Okay, So moving on now to the second key gap that I wanted to address. This is related to Jonathan's question about climate variation. It's not just about mean temperatures. It's also about daily temperature variation, but also climate extremes, climate events that are out of the normal range. So how do these types of extreme weather affect transmission? Here I want to present a case study of how an extreme weather event can affect dengue transmission. This is work done by Mallory Harris, who I already mentioned, who's now a postdoc. And this asks the question, how does extreme weather affect dengue? Specifically, Tropical Cyclone Yaku. This was a storm that hit the coast of Peru in March 2023. This is not a region that's normally very prone to hurricanes. It's not common to have hurricanes in this region, but there was really a major storm and flooding event that happened. So this is showing Cyclone Yaku. Flooding affected over 460,000 people. It left 40,000 people without homes, and 83 people were reported dead in the immediate aftermath of this storm. So it was a huge, major storm event. At the same time, Peru experienced its largest dengue outbreak on record, with 381 dengue-related deaths, which, if you know anything about dengue, it's pretty rare to see a dengue-related death, and it's a pretty huge deal to have this many people dying of dengue. It also was associated with at least 10 times higher caseloads than are usually reported in Peru. So the question is, to what extent was this cyclone driving this unprecedented dengue outbreak? So here we can think about our trait-based model. We can imagine a cyclone is going to create flooding, so it's going to create a lot of vector habitat, maybe increase the abundance of the mosquito. We can also imagine that the storm damage might have increased vector-human contact because people are displaced from their homes are damaged, vector control is probably down in a lot of places that were affected. So these are kind of the mechanistic thinking for why we might expect the cyclone to affect dengue. We can then, again, thinking like an economist, we can leverage this quasi-random natural experiment that happened where this cyclone hit a particular part of the Peruvian coast, it didn't hit other places. Can we leverage that to understand how a cyclone might affect dengue incidence? And to do that, we're using what's called a generalized synthetic controls approach. What this does is it looks at each unit, so each district, where the cyclone hit, and it constructs what we call a synthetic control of other units where the cyclone didn't hit, but if we sort of create a weighted average of those other units, we can match the dengue dynamics prior to the storm. So this synthetic control becomes a control for each affected unit, so they have very similar matching dengue dynamics before the storm, but then the storm hits and they diverge. And the amount of that divergence is our estimated effect of the cyclone. So this is what this looks like in pseudo-equation form. So we have dengue incidence in each of our affected district, or each of our districts in Peru, so 561 districts from 2010 to 2023, which was provided by the Peruvian CDC. And then we have our estimated cyclone effect, which we defined the affected units as the ones that had anomalously high precipitation during this 3-week period when the cyclone was active, or the cyclone and its aftermath. We then compared these to their synthetic controls, which were constructed so that they could match the pre-cyclone dengue dynamics in each of the affected units. And then we're also controlling for temperature and precipitation. So this is now looking at the effect of the cyclone above and beyond the normal effect that just having lots of rain and warm temperatures would have had. So this cyclone also hit during a regional El Nino event, so it was also warmer than average at that time. And then we can add a weighted sum of latent factors. This is conceptually similar to those fixed effects that I talked about in the last model, where you're kind of controlling for a lot of the shared variation that might affect these units so that we can subtract out that more, or we can sort of isolate that more quasi-random variation that's of interest here. So this is what it looks like. So in black, we have our observed average case dynamics in the affected units, the places that were hit by the cyclone. And in red, we have our matched synthetic controls. So you can see that prior to the storm, there's really nice agreement between these two trajectories. And after the storm hit in March 2023, we see this rapid divergence. So this difference is our estimated effect size. We can zoom in on that, and we can estimate that Cyclone Yaku caused about 38,000 dengue cases spread over the next 6 months. So we had significantly elevated dengue cases starting about a week after the storm hit and ranging for 6 months after that. This amounted to about 67% of the total dengue cases in those cyclone-affected units. It was substantially elevated for over 6 months, and it amounts to about 17.8 cases per 1,000 people, just to give you different ways of kind of understanding this number. The other thing we found really interesting is that, oh, something might not be showing up. Okay. You get to trust me on this one. This is a Mac PC conversion situation that I didn't notice before. We actually can estimate a district-level effect size. So each district has its own estimated effect size. And if we plot those according to the average temperature in the post-cyclone period, we see lots of large positive effects when the temperature was above 24 degrees Celsius. And none of those effects were positive when the temperature was less than 24 degrees Celsius. Isn't that convincing? Don't you see it? It really is convincing. I will show it to you sometime. And so that tells us that it's not only the effect of the cyclone, but also the cyclone happening in places where it was warm enough to have dengue transmission. So that kind of underlies our thermal biology thinking as well. We can then do climate attribution and say how much more likely was this extreme climate event because of anthropogenic forcing. So what we're doing here is a pretty simple comparison of CMIP6 models. These are different climate models. In the pre-industrial baseline, the kind of near historic period, and the current period, which we define as starting in 1961. And we say, how frequent were these types of warm, wet events that we observed during the cyclone? And what we see is very clear statistical evidence that these are way more likely because of anthropogenic forcing. In fact, the types of warm, wet conditions that we observed during this cyclone were 324% more likely because of anthropogenic forcing. So we're now able to kind of trace the link between anthropogenic forcing causing climate change and extreme events causing large dengue outbreaks. So extreme weather can drive prolonged surges in dengue, we think, by increasing vector habitat and human exposure to those vectors. Okay, so finally, how will mosquitoes adapt to climate warming? When I show this graph, a lot of times what people start thinking is, well, okay, so you're saying it's going to get way worse. For some of these pathogens, you know, we might start seeing declines in transmission at temperatures as low as 25 degrees Celsius. Is that really going to happen, or are these mosquitoes and parasites just going to adapt to do better at warmer temperatures? And this is, to me, one of the really key empirical gaps in this field. How will these mosquitoes adapt to climate warming? So we've conducted a body of research to try to address this question, led by Lisa Cooper, who was a PhD student in my lab, now a postdoc at Berkeley, Kelsey, who I already introduced, and Johanna Farner, who's a current PhD student in my lab who's just about to finish up. And we first reviewed the literature, and we're surprised to find that there was basically not much evidence at all, one way or the other, about how much adaptive potential mosquitoes might have for climate warming. So there's very limited empirical evidence, but when we look into the kind of evolutionary rescue literature, there are models that we can apply that allow us to predict whether or not a species will be able to evolve in order to track an environmental change. And those models are a function of generation time, population growth rate, how much phenotypic variance they have, how heritable that variance is, how strong the selection is, how much plasticity they have, and how environmentally sensitive the selection is, as well as just how fast is the environment changing. So if we think about mosquitoes, we have a pretty good idea of these two things, and we have basically no idea of the rest of these things. So this really told us that there's a big empirical gap out there in understanding how mosquitoes will adapt to climate warming. So we set out to develop a model system to allow us to close this empirical gap, and that is Aedes syrensis. It is called the western treehole mosquito. It breeds in these water-filled tree holes, so you can see the little wiggly mosquitoes in there. We think it's a good model system for studying temperature adaptation because it has such a broad thermal range in its native environment. It occurs all the way from the border of Canada to the border of Mexico. You can see the temperature gradient that it occurs in, and from the coast into the mountains in California. It's also local to us. It's easy to sample. You can just use a turkey baster to suck up the tree hole water. That's literally how we do it. It's not a major vector of human disease, so it's safe to work with and easy to work with in the lab. So we developed this model system, especially the three people that I just mentioned developed this model system to ask a couple of important questions. First, how much do the life history of traits of this mosquito species vary across this range? And if they vary in response to temperature, is that correlated with their source environment, which would be indicative of local thermal adaptation? So we conducted an experiment where we collected 10 different populations from across 1,200 kilometers of this species range, reared them for a generation in the lab, and then exposed them to different constant temperatures. So each population is experiencing each of these different temperatures, and we're measuring all of its life history traits. This is what we find. So I'm not showing you the data here. I'm showing you the fitted models because the data is just overwhelming to see all that information in one place. But each one of these plots represents a different life history trait versus temperature. Each one of the lines is a different population, colored from blue in the north in Eugene, Oregon, down to red in the south in San Diego, California. if you look across these different lines, you can see that there is variation among populations in their thermal performance curves, but a lot of it is in the vertical direction. So some of them just perform better than others, but they're not necessarily shifted with respect to temperature. And in fact, the only one that's really shifted with respect to temperature is this pupil development rate trait. So the pupae are developing at high temperatures better in some populations than others. So if we zero in on this variation, here now I'm showing that laboratory estimated upper thermal limit for pupil development rate on the y-axis versus a measure of their environmental source temperature on the x-axis. And you can see that there's a strong positive correlation between those, suggestive or at least consistent with local thermal adaptation. We can also see that there's actually pretty substantial variation in this trait. 1.6 degrees Celsius might not sound like a lot, but it's actually 5 times higher than the average for ectotherms based on this meta-analysis that was done by Jennifer Sunday and colleagues. So it actually is quite a bit of thermal variation in this trait. We can also see that there are many days per year that already exceed the thermal limits in their environment. So 35 degrees is above their thermal limit for this trait, and there's multiple days a year in every one of these sites that's above 35 degrees, suggesting that they're already facing really strong selective pressure based on temperature. So to summarize what we found here, mosquitoes are strongly thermally adapted in this one trait, but not necessarily in other traits. And mosquitoes are already facing stressful temperatures in their natural environments. So next we wanted to ask, within a population, how much standing variation is there in acute and chronic thermal heat tolerance, or thermal tolerance? And what is the genetic architecture of this? So this is really trying to help us understand how much adaptive potential there is. How much do these individual mosquitoes vary in their thermal tolerance? So again, we did an experiment. This time we took a genetically diverse population from a single point in the middle of their range in Solano County, California, reared them for 2 generations in constant conditions in the lab, and then we did a control environment where we just reared the mosquitoes at 22 degrees Celsius, which they really like, and a heat-selected environment where we put the larvae at 30 degrees Celsius, which is very stressful to them. Then those that survived, we then reared them for the rest of their life cycle at 22. We then conducted a thermal knockdown assay, which is a measure of acute heat tolerance at the adult stage, where you put them at a high temperature and count how long it takes for them to fall down. And then, this is like a validated thing that people use in the field. And then we did whole genome sequencing on those individuals where we knew their phenotypes. So then we can try to match their genomic variation with their phenotypic variation here. What we find is, as designed, this high temperature selection was very bad for survival for larvae, but there were some larvae that were able to survive it. So not all larvae died from 30 degrees Celsius. So of those individuals that survived these 2 treatments, we then performed the thermal knockdown. And here we see a box plot of their average knockdown time, which is sort of our measure of their acute adult stage thermal tolerance. You can see, oh, my points aren't showing up, so you don't get to see the male versus female variation. But you can see from the box plots there's a little bit of difference, but not really that much difference on average. But there's quite a bit of variation among the individuals. So a lot of individual level phenotypic variation to work with. So then we get to link this back to the genomic architecture of these traits. Specifically, are there just one or a few loci or many loci involved? How are they distributed across the genome? And are these loci the same for both the survival of chronic larval heat exposure and acute heat tolerance at the adult stage traits? So I'll go through this quickly, partly because it's not really my area of expertise and I don't want to say too much, but partly because we're also running out of time. But this is a map showing across the genome of this mosquito. These are the SNPs that had elevated variation in the thermal tolerance phenotype for the chronic larval heat stress. So in other words, the individuals that survived heat stress compared to those that were in the control population. We can see that depending on exactly how you measure it statistically, you have over 100 different SNPs that are associated with this phenotype. And they're across all 3 chromosomes of the genome, but there are some clusters of SNPs, which we'll get back to. In contrast, when we look at the acute adult heat stress phenotype, we see that there's also over 100 SNPs associated with this, but they're much more scattered across the whole genome. If you look statistically, there are more than you would expect shared between these 2 phenotypes, but they're certainly not all the same. You can see quite a bit of variation. And these clusters of SNPs that were associated with larval heat stress really suggested to us that there could be structural variation in the genome that could be involved. And so these are short-read data, so they're not ideal for looking at structural variants, but using short-read sequence data as best we could, we identified signatures of potential chromosomal inversions related to particularly these 3 regions of elevated SNP density. So this is something we're really excited to follow up on, that there might be some structural variation associated with heat tolerance, which has been found in some other mosquito species, structural variation associated with ecological adaptation. There's a few candidate genes that we identified, which are things that you might expect, like heat shock memory, stress response, and DNA repair. And finally, we were able to go back and parameterize that evolutionary rescue model that we proposed in our review paper for this species using all the experimental data from our experiment. So this is the heritability times the phenotypic variance on the x-axis, the strength of selection on the y-axis. The color gradient is the maximum amount of warming that could be tolerated in a given year. The dashed line is our estimated most likely rate of warming that's actually occurring. And then this is kind of our best estimate of where we are in parameter space for this species, suggesting that this species has more than enough variation to be able to adapt to keep pace with warming. Now, we're a little bit uncertain about the maximum population growth rate. So if we look at different levels of maximum population growth rate, we can see that as the growth rate increases, we're more certain that the mosquito is likely to be able to adapt to keep pace with warming, given the amount of genetic variation and phenotypic variation that already exists. Okay, so in this last part, we showed that mosquitoes have thermal adaptive potential. Heat tolerance is variable, both within and between populations of this mosquito species. Heat tolerance is polygenic. There's lots of different SNPs and potentially chromosomal inversions involved that we're excited to follow up on and learn more. I just said that. Evolution is likely to be able to keep pace with warming in this system. We also did some research that I didn't have time to present to you, showing that behavioral thermoregulation is also critical for vector mosquitoes. So in other words, their ability to seek shade is already allowing vectors to escape a lot of the negative consequences of heat. So to bring all this back to this kind of climate change and infectious disease framing, we find that there are nonlinear effects of temperature on vector-borne disease transmission, which lead to, in some cases, directional shifts. So not just expansions, but also shifts in where we see transmission of different diseases. For dengue, it's likely an expansion because the thermal optimum is so high. We also found that these trait-based approaches can be foundational for grounding our hypotheses, particularly for giving us that prediction that we might see nonlinear effects of temperature on transmission, and also giving us predictions about mechanistically how things like extreme weather might affect transmission, which then allow us to layer on complexity in these systems. More specifically, the new research I showed you found that climate warming and extreme weather have already dramatically increased dengue burden, that climate mitigation is critical because it's still not too late, despite the effects that it's already had, to mitigate some of the worst impacts of these climate-driven disease expansions, and that mosquitoes are likely to have adaptive thermal potential. So I want to close by kind of getting back to this idea that humans are transforming the planet. So why are we doing this research? I think one of the things that's really important that we can do is quantify the effects of global change on infectious diseases. The reason that's important, mechanistically understanding it is also important, but being able to actually put numbers, however imperfect those numbers might be, is valuable because it allows us to kind of determine environmental policy while taking health concerns into account. So we can think about things like loss and damages funding and climate negotiations. Right now, that does not account for hardly any impacts of health, let alone infectious disease impacts. We can think about the motivation and rationale for climate mitigation, as well as developing ecological levers for health, whereby we can improve the health of the environment as a way of trying to reduce human infectious disease burden. So with that, I'll stop there, and I'd be happy to take any questions. Thank you. Juan.
Yes. Have you considered the effect of deforestation in
Yes.
the Gaussan model?
Yes, we have. In other ones that I didn't present today, we've looked at deforestation, particularly malaria in the Amazon, where there's a really big effect. It's complicated because it's variable. So like if you're deep in the Amazon where most of the forest is still intact, that's where you see the largest impacts of deforestation because the malaria vector in the Amazon is really forest edge associated. So when you do that early stage of deforestation, you create a lot of forest edge and you have a lot of people living and working at the forest edge. If you move into the outer part of the Amazon where most of the forest is already gone, deforestation only pushes that forest further away, so it has less of an effect. It's a little trickier with deforestation for these econometric assumptions of like plausible random assignment. Like it's a little harder to deal with. So sometimes you have to use additional tools like instrumental variables that help you to try to like better approximate that random assignment assumption. But definitely, I think deforestation has a big effect, especially on diseases that have a more zoonotic or sylvatic lifestyle, where they use forest-associated mosquitoes and hosts. I think deforestation is having a big effect on those diseases. We'll go here first, and then Indra next. Yeah.
Just like a follow-up question about
Yeah.
that, because I was wondering, there are a few countries in Latin America that have been pushing for reforestation efforts.
Yeah.
So I was wondering, like, can you sort of validate the impact of reforestation in the singular diet?
Yeah, that's a really good question. We've had a couple of actually undergrad thesis projects in my lab recently using reforestation. Brazil has this really cool data set called Map Biomas that has lots of different land use data, including reforestation data. And we've looked at that as a predictor of leptospirosis and leishmaniasis, and so far haven't seen really strong relationships, but I think it's pretty sparse still, so like it's a little hard to tell. And it's always unclear to me when you say reforestation, like, do you get back the thing that was there before? Like, probably not exactly. You're probably missing a lot of the forest. One of my colleagues studies what she calls silent forests, where the trees come back, but you don't necessarily have the large animals and the other species that might contribute to the ecosystem. So when you're thinking about something like Oropuche or leishmaniasis or leptospirosis, where there's zoonotic hosts, those types of effects are going to be really important. So I think it's not necessarily true that reforestation would kind of reverse the damage of deforestation. Indra?
Thank you so much. That was super interesting.
Thank you.
I was wondering, beyond like the growth of the mosquitoes themselves, if you looked at like how often they need to feed, like if the metabolism like
Yeah.
accelerated or something, which is like potential transmission risk.
Yeah, that's definitely one of the factors that's incorporated into the R0 model, where their biting rate, that's the reason that their biting rate is temperature sensitive, because basically they metabolize the blood faster and they feed faster as it gets warmer, up to a point, and then it turns over. Sometimes people don't believe me that it turns over, but it actually, empirically, you do see that biting rate turn over at very high temperatures.
Is it
Yeah.
because they die?
They do die at a higher rate, but they also just stop blood feeding when it gets really hot. They just are not, things are not good for a mosquito. Like, you get to these temperatures where they only survive a couple of hours at that temperature, and they're just not really doing well physiologically. So you see like lower blood feeding at those temperatures also. But it gets harder and harder to measure also empirically, because your sample sizes get small because all the mosquitoes are dying. So, yeah. There is that, yes. Did I see a question over here? Yes.
I guess kind of on that note, just a technical question that I just wanted to ask you.
Sure.
How do you decouple, like, once it gets really hot, people are also like not going outside as much probably, right? So then how do you decouple the human activity aspect to
Yeah.
the other things
Yeah.
you're measuring, like with rain or something?
Yeah.
Wash out mosquitoes or make it harder for them to fly or something.
Right.
And then how does that come into the model?
Yeah, with the temperature effects, I think there's a really clear physiological effect that we can measure experimentally. And so the fact that I'm not doubting that those things also happen, like people might be less active or whatever, but also there's just a direct physiological explanation for the fact that the transmission starts to turn over at high temperatures. Also, like when we first started publishing this work, because our model predicts that malaria optimal temperature is 25 degrees Celsius, and there's pretty clear evidence from the field that that's actually true once you look. But people just didn't believe us, and they said, "Oh no, it's just because it gets too dry. It's too. There's not enough rain. It's not driven by temperature." But sorry about that. But there really is direct physiological effects of high temperatures that are negative for the mosquito as well. So in these kind of econometric modeling approaches, it's all sort of like baked in there, like the behavioral and the direct physiological effects. In this case, they're maybe going in the same direction. So like temperature is reducing, maybe. Although Indra and I were talking about how, oh yeah, in Puerto Rico it's so hot, we're always outside. So, you know, that it could go in both directions there. But there certainly are behavioral aspects that, like, these econometric models sort of bake all of that in. So to whatever degree that behavior is strongly correlated with temperature, you're going to see that as part of your effect. Yeah, great question. Yes?
I like that trait-based approach for thinking about the effects of climate change. It makes a lot of sense for grounding your hypotheses. To what extent is that the dominant approach, and what are the pros and cons of taking, trying to generate these mechanistic hypotheses versus something where you don't even know all of the potential ways this could play out, and you might miss something?
Yeah, thank you. That's something that I'm like learning about and thinking about all the time, because with an R-naught model, like it's easy to believe an R-naught model parameterizes the function of temperature. It kind of has this level of abstraction that's like, okay, sure, temperature's affecting the mosquitoes, but what else is happening? One of the most common things people wonder about is how susceptibility dynamics are coming into play, particularly when you have an endemic disease that's been circulating for a long time. Like, well, maybe warming won't really make it worse because there's already herd immunity or something, but which I, for a variety of reasons, don't necessarily buy that. But some people do SIR type modeling or SEIR modeling that then incorporates that, the dynamics of susceptibility, as well as maybe a climate-forced transmission parameter or something. And that can be a really powerful approach, but it can also be really difficult to apply at large scales like this, because if you've ever tried to fit one of those models for a vector-borne disease, it's like painstaking to get them to really match up with the true dynamics. And to do that in many different places over time is just like kind of a technical challenge that hasn't been solved yet. And so one thing that we've struggled, like in reviewers with this climate warming and dengue study are always asking, how can you really trust these projections going forward? And so we've done lots of different ways to try to especially account for that susceptibility issue. Like, for example, for the places where we have really long time series, we can compare the estimated effect size at the end of the time series versus the beginning, where hypothetically susceptibility should have changed in those time periods, and we find that we can estimate the same effect. We can simulate an SIR model that's climate forced and still recover the effect. So we think it's pretty robust to these types of things, but I think if you really want like detailed prediction that's mechanistic, then I think like there's really no substitute for fitting an SIR model to a location. But that just requires a lot of, like, it can really only be done at kind of a very local scale with very detailed data if you want it to work nicely. So I think compared to some of the respiratory diseases where like the interplay between SIR models and climate change has been a little easier because it's easier to fit those SIR models, like I think vector-borne disease hasn't used SIR models quite as much at that large scale. Yeah. Yes.
There's a question online.
Oh, okay.
But before I do that, I just want to say that for people who want to leave at 2, they can, but we have a few more minutes for those who want to stay. So the question says, you noted that projections didn't account for changing disease control measures. Has there been work on the relative costs of climate mitigation versus adaptation in terms of investing more in implementing vector control?
Well, we're already not doing a very good job with vector control. Like you saw that pattern of like, this exponential growth in dengue over the last few years. And so I don't have great faith that we'll just like click it into gear and get the vector control. I mean, there are some new promising tools on the horizon, some dengue vaccines also promising, but I don't have a lot of confidence that we'll be able to not only, make a dent such that you could reduce transmission compared to what it is now, but also overcome the additional force of infection that's coming from the warming climate. So I think like it would be great if some of these innovations in vector control and human vaccines would make this completely obsolete and not useful, but I think that's probably unlikely. But I would love to be proved wrong on that. Because it's just a very difficult vector to control because it breeds in really tiny amounts of water, like a bottle cap filled with rainwater can be an Aedes aegypti habitat. So it's just very hard to like find every last one and kill them. That's why things like sterile male mosquito releases and Wolbachia and other types of genetic control mechanisms are appealing, because mosquitoes are much better at finding each other than we are at finding mosquitoes. So I actually just read a news article that some parts of LA, they're starting to deploy sterile male mosquitoes. And they're asking voters if they're willing to approve an extra $20 a year property tax to support this. I'm like very curious to see how that's going to play out in LA. But it's, people are trying it. It's not an easy thing to deploy at a very large scale, though. Yeah. Another question?
So I work on bacterial evolution, and bacteria kind of easy because they like divide into and then live or die. But mosquitoes clearly have different stages.
Yeah.
So I was wondering if selection at different stages has different adaptive potential. Like if you were going what the different selective pressures
I would love to do more on that. Like how strong is the selection? Or what are the drivers of selection at the different life stages? How strong is it? Like what's the prevailing factor that's driving? Or are there tensions between selection at different life stages? The other thing, when I found out a couple hours ago that this talk was only 35 minutes instead of 50, which I kind of almost pulled off, the thing that I cut was this really cool host-parasite interaction that this treehole mosquito has, where when it's a larva, there are ciliates that live in the water, and it eats all those ciliates. It's a predator on these prey. So the ciliate has evolved to become a parasite of the larval mosquito. So if it detects that the mosquito is there, it irreversibly becomes a parasite, attacks the larval mosquito, and kills it. And it's really cool. And it's really temperature sensitive also. It has this nice nonlinear effective temperature. We can measure it in the lab. It perfectly lines up with what we observe in the field. And the parasite is locally adapted to the mosquito. So they do much better infecting their local sympatric mosquitoes compared to allopatric. So that's just one example of a really strong selective pressure on both the mosquito and the parasite that occurs only at the larval stage. And most of the infected mosquitoes get killed, but if they don't get killed, they're parasitically castrated so that they lay ciliates instead of eggs. So it's just I really, anyway, you can see the rabbit hole I've just gone down. So that's something that we're studying. That's part of the reason we're so excited about the tree hole study system, because I think it allows us to really think about the ecology really holistically. What are the different selective pressures on this mosquito's life cycle? It's already dealing with a lot of temperature variation in its environment. Maybe it'll just shift its seasonality a little bit earlier and no problem. So is temperature an important selective pressure compared to these other things? I think that's a really big open question. And even just the selective pressure of temperature across the life cycle. Certain traits have cooler thermal limits than others. So there might be some that are more strongly selected. It's also this really seasonal life cycle where, in California, it only rains in the winter and spring. And so they have this kind of synchronized life cycle where the larvae all hatch in the winter and spring, and then they develop, and then the adults come out in the late spring and summer, and then they lay their eggs and die. And then there's no mosquitoes until the next rain. So the temperature they're experiencing is different at different parts of the life cycle too. I think there's just a huge amount of research that needs to be done. And also on the actual vector species, not just on our cute little California tree hole mosquito. So yeah, open question, but a very, very good one. Okay. Oh, one more. Okay.
Sorry. What's the conversation around how Wolbachia mosquitoes change with climate change?
Yeah.
Will they fare better?
Yeah. So I haven't done this research directly myself, but a postdoc in my lab, Valerie Vasquez, did research on that for her for her PhD before coming to postdoc with me. One of the things, in the lab, they had observed that Wolbachia infection was sometimes lost at high temperatures, which is a concern. But they did some modeling, really life history, population dynamic modeling of heat waves, and found that actually they think it's likely to be pretty robust to heat waves that we're going to experience. So at least that dimension of Wolbachia seems pretty robust, which is great. And, Wolbachia, it's expensive to deploy. It's kind of a cool high-tech solution, but it has the advantage that it's very limited in any off-target effects it could have compared to an insecticide or something. And it also, there's what we call population replacement versus suppression strategies. And the population replacement strategies, you still have a mosquito population. It's just no longer able to transmit a pathogen. So there's maybe less selective pressure to evade Wolbachia infection. Whereas the suppression strategies drive the mosquito population down. So then you're going to have selection for escaping that. So a lot of interesting ecological and evolutionary complexity to think about with those strategies. But anyway, that's not really directly my work, but Valerie Vasquez has great work on that. So, yeah. Well, I know you're probably all ready to go. So thank you all for your attention and for inviting me.